A muscle fiber is a cylindrical, multinucleated cell composed of microfibrils that is contracted when stimulated. Huxley and Hansen proposed a sliding filament model. They hypothesized that banding patterns in the sarcomere are actually caused by two types of long filaments, thick filaments and thin filaments, and that these filaments slide past one another during a contraction. Thin filaments are composed of two coiled chains of globular protein called actin. The thick filaments are composed of multi-strands of long proteins called myosin and are anchored to the middle of the sarcomere. During contraction, the myosin thick filaments grab onto the actin thin filaments by forming cross bridges. The thick filaments pull the thin filaments past them, making the sarcomere shorter. In a muscle fiber, the signal for contraction is synchronized over the entire fiber so that all the microfibrils that make up the sarcomere shorten simultaneously. Now that we've covered how muscles contract, we can now look at how muscles grow and repair themselves. A study from the Neuromuscular System series shows that muscle biopsies of experienced bodybuilders have shown that it is the size of the individual fibers within their muscles that was responsible for their abnormal muscle size, and not actually the number of fibers present. Hypertrophy is thought to be the main reason behind muscle growth. There are two types of hypertrophy: sarcoplasmic hypertrophy, which is the increased volume of tissue that supplies energy to the muscle or is involved with the neural drive, and sarcomere hypertrophy, which increases the volume of contractile machinery. Sarcoplasmic hypertrophy occurs when higher rep ranges are used. This is often referred to as a pump in many bodybuilders. This also increases the concentration of enzymes involved in the oxidative phosphorylation and anaerobic glycolysis mechanisms of energy production, and increased in volume of sarcoplasmic fluid inside the cell, and also between the actual cells. Sarcomere hypertrophy is the increased volume of contractile machinery. It makes up nearly 80% of the muscle. When a muscle fiber develops sufficient tension for sufficient time, increased fatigue impairs the actin-myosin cross-bridge cycling necessary for contractile filaments to maintain force production. This impaired cross-bridge cycling under load results in trauma to the contractile filaments, as some cross-bridges are subject to tension greater than they can structurally support. Additionally, training leads to post-workout breaches in plasma membrane integrity that allows calcium to leak into the muscle cells. This intracellular increase of calcium levels activates enzymes called kalpains, which remove pieces of damaged contractile filaments. After a muscle has been broken down, it needs to be built back up by stem cells called satellite cells, or myoblast. Satellite cells work by increasing the number of satellite cells. They have to become more like muscle cells, and then they fuse with needy muscle cells. After this process, growth factors are released. These growth factors can leave and go in and out of the surrounding area because the muscle permeability has been increased due to damage during contraction. Once outside the muscle cell, these growth factors can cause the satellite cells to proliferate and differentiate. These growth factors promote protein synthesis such as insulin growth factor 1, growth hormone, testosterone, and these commence the growth process. Protein synthesis occurs because a genetically coded substance called messenger RNA is sent out from the nucleus to the ribosomes. The nucleus is believed to release increased mRNA in response to tension or microfibrillar damage done by the result of insufficient cycling of actin-myosin cross bridges during the intense muscular contractions.